evening, everybody. We have a very interesting speaker this evening. Came to us from Mike Farley. He has been working with Mike on identifying some of the animals that they're seeing on the game cameras out at Berry Springs. So our speaker tonight comes to us from Tarleton State University, Dr. Russell Pfau. He has a master's degree in biology from Midwestern State University, a PhD in zoology from Oklahoma State University, and he's been a faculty member out at the Biological Science Department at Tarleton since 2000. He's currently teaching courses in genetics, ecology, and evolution. His research focuses on population genetics with several studies related to conservation of threatened species. So, with no further ado, Dr. Pfau.
All right, so species are sort of the currency of naturalists. If we didn't have money, we wouldn't have accountants, and if we didn't have species, we probably wouldn't have naturalists. So what I want to focus on here is what species actually are, and to some people it's obvious, and to others it might not be. And so my approach here is going to be to sort of start out talking about some issues related to species. I'm going to wait until the very last slide to give you my personal definition of what a species is. And then on the way, I'm going to share with you my research and how my research has been answering the question of, so what is a species? Okay, so there's two ways to approach what species are. One is from just a practical approach, and the other is theoretical. People have gone and talked with the native peoples of South America, living in the tropical rainforests, and asked them, how many different kinds of birds are there in your neighborhood here? And they would say, they'd give a number. And it turns out to be almost the exact same number as scientists recognize. So the native peoples of South America recognize the same number of species in that area as scientists do. So it's pretty remarkable. But then you ask them how many different kinds of ants there are. And they'll say, well, there's two. When in reality, the scientists recognize 800 different species of ants in that area. So for some species, everybody kind of recognizes species in the same way, but for others, not so much. And it probably has to do with how important those organisms are. So maybe the native peoples, you've got the ants that can hurt you and the ants that don't, and that's all they need to know. They don't need to go into any more detail than that. Whereas the birds, you might have different species for different decorations, rituals, or food sources, that sort of thing. So from a practical standpoint, we want to know, well, how can we recognize one species and distinguish them from another? On the theoretical side, what things in biology can be called species from a purely theoretical standpoint, without going out there and looking around to see what you can recognize? And with both of these aspects, there's often a considerable amount of disagreement among scientists. And that's one of the things I'm going to get into, is that disagreement, the source of that disagreement, and why it's really not as easy as what you might think to recognize species. And I don't know if I can see what I normally do here when I'm face-to-face is I ask people how many species you see. So I can't see people's responses or anything like that from my view at the moment, but just be thinking to yourself, how many species do you see there? I'll give you a second. And if y'all know how to type in a response and other people can see the response, feel free to share that with me. All right, time's up. There's just one. So all of those individuals are members of the same species. They're Asian lady beetles, an introduced species. Unfortunately, that's really common now here in our area. Harmonia axuritis. And there's a lot of variation from individual to individual. Why is there so much variation within this one species? Why do they all look so different from one another? Well, there's a proximate explanation and an ultimate explanation for that. The proximate explanation is that, well, there's variation in the genes that affect their coloration. The ultimate explanation would need to answer, well, why does that variation exist? Does it have a purpose? Is it just random? Those sorts of questions are very hard to address. We just don't know why there's so much variation among individuals of this one species. All right, here's another example. These are two species of butterfly. And I'm not going to attempt to pronounce those. I'm usually pretty good at pronouncing scientific names, but the top one there is kind of bluish color, and that was described in 1911. The bottom one was described a year later, and it's kind of a brownish color. Very different, easily recognizable. All of one species is blue like that, and all of the other species is brown like that. But it turned out to be wrong. What they had done is describe the male and the female as different species. In fact, that's just one species, differences between the sexes. And one more example here, the red-bellied pita, one species. Until very recently, and now scientists recognize 12 different species of these things. And you can look at the little circle of the painted images and see there's some slight variation in color, and they occur in different geographic locations. So every little geographic location has a different species, which is very slightly different from one another. Only recently recognized as 12 species. Now, taxonomy is the scientific practice of naming and categorizing living organisms. Humans have been doing taxonomy informally forever, as long as humans have had a language, and those are common names. For example, on the left-hand side there, we have a right side, we have a ground squirrel, and there in the middle, we have a salamander. The problem with common names, however, is that they vary a lot from one geographic region to another. So here in Texas, those burrowing mammals that throw up mounds of dirt are called gophers. If you went to Florida, those little burrowing mammals that throw up mounds of dirt are called salamanders. Salamanders, gophers look very different. Gophers in Florida, salamanders. And we think that the name salamander for gopher came from the phrase sandy mounders, because they live in sand and throw up mounds of sand. And say, if you're living in the backwoods of Florida and you say sandy mounders real fast, it kind of comes out maybe as salamanders. And so probably the language evolved from sandy mounders to salamanders. Now, if you go up to North Dakota, the ground squirrel on the right-hand side, they call those gophers. So obviously people from Florida, North Dakota, and Texas cannot communicate with one another about these organisms without getting thoroughly confused. So scientists don't use common names. We use the scientific names. And species have a two-part name, a generic epithet, which is the first name, and a specific epithet, which is the second name. So the gopher, no matter who you are on the planet, who you're talking to, Geomys bursarius, everybody will know what you're talking about as long as everybody's familiar with them. And then the salamander there and the ground squirrel have their own unique scientific names. So the scientific names do not vary from country to country or place to place like common names do. Now, like a lot of things in life, if you don't have rules, then things kind of go haywire. And so taxonomy follows the rules of zoological nomenclature. And you definitely don't want me to go into the details of this. This is extremely boring and dry. I'm just showing you this so that you know that scientists can't just name things. There's a certain way you have to go about doing it, certain rules you have to follow. And when those rules are broken, there's an international commission that advises and arbitrates disputes regarding the naming of things. All right, so these are just names. Why does it matter how you name things? That sort of, does it matter that we worry so much about species are and that sort of thing? There's three main reasons, and one, you can't communicate with one another without names. The names need to accurately reflect reality. So reality is the diversity of life out there, and the names we put on those little bundles of diversity need to be somewhat accurate when we communicate with one another. And if we don't accurately name these things and recognize them as unique biological entities, then we don't really know what we're studying. And to know what to protect. Endangered species, threatened species, if you haven't correctly identified the species, then you're going to have a harder time knowing what it is that you need to be protecting exactly. And there have been several notable disputes when it comes to endangered species about, well, is that actually a species that needs to be protected, or is it not two different species that need to be protected? Okay, so sort of getting at the idea of what species are. This is not really a definition of species. I'm going to have something a little more specific there at the end, but for now, species are groups of organisms, groups of individuals that have similar characteristics. That's not good enough for scientific purposes. If you think back to the three examples I gave you of the bird, the other two examples, can you remember now what they are? The ladybugs and so forth. Just noting that they have similar characteristics isn't enough. But what kind of characteristics do we need to base our decisions on what species are? And how dissimilar can individuals of the same species be? If you just think of humans, Homo sapiens, we range from like 8 feet tall to like 3 feet tall. Differences in hair and skin color and eye color and all sorts of characteristics, but we're all members of the same species. So how different can one be, but be a member of the same species? And that's really where things get tough. And that's sort of what this presentation is about. There's different opinions. And because of that, we have what is known in the scientific community as the species problem. And people have been debating what species are and how to recognize them for over 160 years. And the debate hasn't slowed down over that period of time, and there hasn't really been a good consensus. In other words, you can still have a few beers at a scientific conference and argue over what a species is. Okay, now we have species concepts, what are species, and species delimitation, how do we recognize them? So how do we identify different species? That's species delimitation, that's our practical side of things. And then what causes different species to exist in the first place? That's our species concepts, that's the theoretical side of things. Last time I looked, there's at least 26 unique species concepts or delimitation criteria that scientists have published. So there's at least 26, probably up to around 30, 35 or so, different ways of defining what a species is. And every so often a scientist will think, well, I've got it figured out, and he'll publish a new concept of what a species is. Most, or not most, many of those 26 unique concepts of species are similar, almost identical, and some are more different than others, so they can be kind of categorized. So we can categorize species concepts kind of like we categorize species themselves. And I'm not going to read any of this, and y'all don't have to read any of this either. I'm just showing you this table so that you can see on the left-hand column, these are the names of different species concepts that scientists have proposed. And then in the middle, different kinds of properties that define each type of species. And then the authors that have either come up with this concept or advocate for it there on the right-hand column. So this goes all the way back to the 1940s when people first started formally thinking about, well, gosh, what do we mean when we say species? So Wright, Mayer, and Dobzhansky were the early workers in just trying to define the properties that we use to think of what species are. And even as early as 1995 and 2001, 1999, people have still been proposing new concepts. All right, so properties of species. What are the properties of species? What do we need to look for to say this is one species and that is another species? Well, I'm just going to talk about four of them. There's more than this, but these are the four main ones. They look different. So obviously, an elephant and a giraffe are different species. Nobody's going to argue with that. They look different enough. That does the trick. That's really all we need there. But we have species that look identical or very similar to one another. Yet they cannot reproduce with one another. And this is because of something internal to the organism, not because they're geographically isolated. But if you have two kinds of organisms living together, interacting with one another, but they do not reproduce because of their own intrinsic physiological genetic differences, then we'd say, yeah, those are different species, even though they may look alike. Occupy different niches. So a niche is just the combination of the environmental factors that an organism needs to survive And so you can have different groups of organisms that need different ecological variables to be able to survive. One might need a drier environment, one might need a wetter environment, that sort of thing. So if they occupy different niches, we can recognize them as being, there's something different about them. Maybe that needs to be recognized as a species. And this fourth one is a little more complicated here. And the next slide is going to help me explain what I mean by monophyletic. But basically, monophyletic just means descended from a shared ancestor. Again, the next slide will help me explain that. Must a species have all of these properties? So does every species have all of these properties? Well, let's look at elephants and giraffes. They look different, they cannot reproduce with one another, they live in different environments, and they are monophyletic, descended from shared ancestors. Each one is descended from a separate shared ancestor. But if we look at the Asian elephant and the African elephant, well, they look a lot alike, but they're a little bit different in appearance. Can they reproduce? I don't know. I don't think they come into contact naturally, so it's hard to know if they would reproduce in the wild or not. They do sort of live in different niches in their monophyletic. So that one fits 3, maybe 4, but we're not sure about that fourth one. And then you have things like cotton rats. Cotton rats are the more common rodent living around here. The ones in the east look identical to the ones in the west. They're capable of reproducing with one another. They don't really occupy different niches, but that fourth criteria, you can check that one off. They do fit that criteria. So gosh, should we recognize 2 different kind of cotton rats? That's one of the examples I'll get into here in a minute. All right, so because I'm going to be showing you some phylogenetic trees, these tree diagrams as part of my research, I want to make sure that everybody has a chance to understand what they mean. So the tips of this tree-like diagram, envision this as just being like an oak tree or a mesquite tree, and you have the very tips of the branches. That's the most recent. If it's a real tree, that's the most recent thing to bud out. And then at the bottom, you have sort of the root of the tree, and that's the oldest part of the tree. And so as a tree grows, a bud might split in 2 and form 2 branches. And then that one branch would have a bud split in 2 and form 2 branches. So the tree branches out as time elapses, with the newest things being here at the top and the oldest down at the bottom. And the connections show how they are related. So 2 branch in a tree, and 2 species share a common ancestral species as well. Okay, so we have different colored blocks here. If you have branches that connect together onto one branch, like here in the red square, all of those branches connect back together as you go down the tree onto one branch. Over here, the blue ones, all of those branches here connect to get together down the tree onto a single branch. So the red and the blue are monophyletic. The branch from which they branch off from represents a shared ancestor, a common ancestor from which those branches were derived. The green is not a monophyletic group because it excludes the blue. So if you were to use monophyly as a criteria for species, we would probably want to choose 2 species here, the red one and the blue plus green group of that species, blue plus green, because they stem from one branch down at the bottom, and then the red because they stem from one branch. Okay. Now, when you go out and explore biological diversity, you see individuals that are identical to one another. You see individuals that are a little bit different from one another. And you see individual organisms that are quite a bit different from one another. And then you see organisms that are really, really, really different from one another. And this represents sort of a continuum. So we divide that continuum up into these false categories. Most categories are false, but a population is a group of organisms that are very similar to one another. Variety, these are organisms, different varieties are more different from one another than different populations. Different subspecies are more different from one another than different varieties, and different species are more different from one another than are different subspecies. And this is a continuum. There's no defining moment at which you can distinguish between any two of these. There's no definition where you can distinguish between population and variety. It kind of just integrates. I'm going to explain why here in a second. All right, so how do species arise? This little fork diagram here is representing how a species splits into two species, species A and B. So we have the ancestral species at the bottom, and then we have two species at the top. And so the vertical axis here on this diagram represents the passage of time. All right, so we have our ancestral species, and then at some point in time, there was a barrier to gene flow, maybe a canyon eroded. For example, there's squirrels living both sides of the Grand Canyon, the North Rim squirrels and the South Rim squirrels. At one point in the past, there wasn't a canyon there, and those squirrels were all just one big happy squirrel species, and now there's two species. So there can be all kinds of barriers to gene flow. Continents split and move apart, and oceans separate them. Deserts form, separating two grassland areas or two forest areas, things like that. Anything in the environment that changes, that prevents organisms from exchanging their genes when they reproduce. Basically, it prevents them from reproducing. And mutations accumulate in each of these isolated populations. So if you have two populations now, at this point in time, one population will acquire mutations. The other population will acquire mutations. Mutations just being changes in the DNA. But those mutations are going to be different. Both populations will acquire different mutations, unique mutations. And those mutations accumulate over every generation. For example, in humans, each one of you has, on average, about 50 brand new mutations that neither parents have, mutations that occur at some point between your parents and you. And if you have kids, your kids have 50 brand new mutations that you don't have. So every generation passes on about 50 brand new mutations. Those accumulate. And so over time, our two populations will become more and more and more and more different from one another. All right, on the right-hand side, we have evidence that this I know there's some technical terms here. If you don't understand a word, it's not really important. Here, the frequency of genetic variance changes. For example, a version of a gene for blue eyes and a version of a gene for brown eyes let's say everybody has brown eyes. There's a mutation with one person with blue eyes, and then over time, the frequency of blue eyes increases. So that maybe eventually 80% of the individuals have blue eyes. That's what we're talking about here when we talk about different allele frequencies. And then at some point later in time, the populations become morphologically distinct. They look different. And then at a later point in time, they come to occupy different niches, and so they have a different habitat. A little bit later in time, they become reproductively isolated. All of these changes here on the right, the biological evidence, all of those things are caused by mutations. So different mutations cause different consequences. So some people focus on morphology, and so they would follow the morphological species concept. They have to look different before we call them different species. But some people would say, well, that's not really all that important. What's most important is they're reproductively isolated. So they would follow the biological species concept. And some would say, well, that's really not that important. It's their habitats, the niches they occupy. That's really what's important in nature. They follow the ecological species concept. So we have these different concepts because different scientists think some things, some properties of species are more important than others. The order in which these properties arise is going to be different for different species. They don't always occur in the order listed in this diagram. That's just one possibility. They might become reproductively isolated before they become morphologically unique, for example. All right. So what I'm saying here is that Mother Nature created the species problem. The species problem is not just because people like to disagree over a beer. The species problem exists because of the way that species arise over time. Different species diverged at different points in the past. Okay, so for two populations that have only recently become separated, there hasn't been much time for mutations to accumulate. But for two groups of organisms that maybe they diverged 2 million years ago, well, that's enough time for quite a few mutations to accumulate. And then you might have two groups of organisms that diverged 20 million years ago. And that's a lot of time for a lot of mutations to accumulate. So they're going to be really different from one another. And so different groups of organisms diverged at different times in the past. Therefore, we have this continuum of degrees of difference between them. All right, so here's where I'm going to kind of change gears and talk about my, my research, my particular research, and how it relates to species. The research is where the rubber meets the road. That's where we discover patterns in biological diversity. That's where we gather the data to try to figure out what species are and that sort of thing. So my research sort of focuses on the genetics of populations and how those populations have evolved over time. And much of my work centers around the population-species interface. Some of the organisms I study are populations of the same species, and some are closely related species. And some, I got no idea what they are. So I don't do a whole lot of work with morphology or reproduction. Most of my work focuses on the DNA, the genetics side of things. I do occasionally try to tie in some morphology, but not a whole lot. I get my DNA from two places within the organism's cell, shown there in red, the nucleus and mitochondria. Both of those places inside the cell contain different DNA. The mitochondria has little tiny circular DNAs, like little tiny rubber bands, where the DNA is in a circle. There in the nucleus, the DNA is like strings. They have ends. And there's all kinds of different aspects about them, but they're two different types of DNA in the same cell of almost every organism. So what I do, I collect the organisms I'm interested in, obtain a piece of tissue, maybe muscle or liver, or if it's in an insect, I might take a leg from the insect, smash it up, expose it to some chemicals that help to separate the DNA from everything else. So at this point, I've got a tube that contains, to the naked eye, it just looks like water. It's just a clear fluid. It looks just like, it's mostly water, but there's DNA dissolved in the water, just like sugar is dissolved in the water. At this point, once I've extracted the DNA, I just have pure DNA dissolved in the water. And then I obtain the DNA sequence. So GAA-TTAAC, the sequence of G's, A's, T's, and C's that make up the little subunits of the DNA. I obtain the DNA sequence from the nucleus and also from the mitochondria, because both of them paint a different picture. They give a different view. And here's one reason why. We inherit our mitochondrial DNA only from our mothers, not our father. So the DNA that is in your mitochondria came from your mama. The DNA that is in your nucleus, half of it came from your mom, half of it came from your dad. So because of that, we can learn different things from those two different types of DNA. The hispid cotton rat was the first organism that I studied as a young geneticist. So this was for my PhD research. Going back 1 degree, my master's research, I was looking at fossils, mainly the teeth of small mammals from a fossil site. So I moved from paleontology into genetics for my PhD. So the little cotton rat here, Sigmodon hispidus, was the first organism that I extracted DNA from. And I've worked on them for several years now. Let's see, when was this? This was probably around this would have been late 1990s. At that point, some other people had said that the eastern cotton rats, they're genetically different from the western ones. And I thought that was pretty cool. And I got some clues from other people that the two contact one another geographically in East Texas. So me and a grad student and a couple of undergrads, we got our rat traps together and went out and ran a transect. And that's what the bottom right diagram, each one of those 10 locations is a trapping location. So we ran a transect across where we thought the eastern and western cotton rats met. And then we came back, extracted their DNA and did some fancy computer work and got this really weird diagram that's really hard to explain, but I'll do my best. Each vertical bar is the genetic makeup of one cotton rat. So we have a bunch of individuals and you can see their locations there. So we have site 1 through 10. We got Kansas, starting on the left, we got Kansas, Oklahoma, Texas, and then the 10 sites, and then Louisiana and Georgia from left to right. If it's solid gray, that means it's Western, the Western form, genetic form. If it's solid white, that means it's the Eastern genetic form. And then these the backslashes here, the hash marks, those represent hybrids. So in the middle of this, these two genetically different types of cotton rats are hybridizing with one another. So over about a 125-mile stretch. So they're mating with one another like they don't care. So, and then the mitochondrial DNA is shown at the bottom. So the gray bar at the bottom, that's showing they're the Western form, and then the white is showing they're the Eastern form. And that transition occurs there in sats 4 through 7. So two genetically different animals, they look identical, same habitat, everything's the same about them except genetics. And they mate with one another. So are those different species? I still don't know. It's been about 15 years since I did this, and I still don't know if I should call them different species or not. So moving on from that, because I'm not going to solve that problem tonight. Gophers. I'm really, really interested in gophers because they only live in sandy soils, and sandy soils become isolated. And so we have all these different populations of gophers that are isolated from one another by soil that they cannot travel through. Out in New Mexico, West Texas, there's 2 subspecies that have been defined by morphology, the one in red and one in green. And so we went in, collected some specimens, extracted the DNA, and created this phylogenetic tree. Again, the green is one subspecies, the orange is the other subspecies. And you can see all of the members of one species grouped together on a single branch here. And all of the individuals of the other subspecies grouped together on a branch there. They're monophyletic. They're very different genetically from one another. They form distinct genetic groups. One of the concepts, one of the species concepts applies here. So I could elevate these to species if I wanted to. These don't overlap. There's a geographic gap where they do not occur. They never interact with one another in the wild. We have no earthly idea if they would hybridize with one another or not. So they're slightly different morphologically, very different genetically. Should we recognize them as a different species or leave them as subspecies? Gosh, I don't know. I'm often sitting on the fence of issues in my everyday life as well as my research life as well. This is showing the nuclear genome. So each little symbol in this graph is representing one individual gopher. And the further apart the symbols are on the X and Y axis, the more different they are genetically. So you can see they form 3 clusters. The green cluster is one subspecies. The orange cluster is the other subspecies. But look here, the orange subspecies forms 2 different groups, 2 different populations that are genetically different from one another. Is there 3 species now? I don't know. And then my last research example here is bumblebees. I've only recently become really, really interested in bumblebees. So look at these 2 bumblebees here. They look different, right? Everybody would probably agree that they look a little bit different. Scientists back in the 1800s, 1800s, classified these as 2 different species. Now we know that the one on the left is a male and the one on the right is a female. And on iNaturalist, this is one of the biggest points of confusion among the bumblebees, is people think that these are 2 different species. So it's still fooling people. because the males look considerably different than the females. So this is a Bombus pennsylvanicus, the bumblebee that is really common in the Georgetown area. So here's an excerpt from a 300-page paper written in 1912 by Franklin. So Franklin in 1912 wrote a 300-page paper on the bumblebees of the New World. And it's really interesting to read things written over 100 years ago. They use a different language, different way of thinking and everything. It's really fun. So just reading the first couple of sentences there. The male of this species, long known as Cytheris elatus, has probably caused as much discussion as any bumblebee in the Western Hemisphere. And so only recently, only a few years before 1912, did people start to recognize that that's the same species. And they used characteristics of the genitalia and body structure and habitat and social interactions to figure that out. That species, Bombus pennsylvanicus, was recognized by de Geer in 1773. In 1873, 100 years later, a different species was recognized, Bombus sonorus. And so the pictures of those are up on the top right. And if you have a sharp eye, you can probably see those look a little bit different. The yellow banding patterns have a different pattern. And the one on the left is a brighter, more vibrant yellow than the one on the right. Right? So you can see here between the wings, this one has a yellow band. Here between the wings, it's black. Two different species, recognized 100 years apart from one another. Franklin in 1912 recognized these as distinct species. Peters in 1968 argued that they're the same species, that they're different subspecies. Millerone in 1971 agreed that these are two varieties of the same species. Thorpe, 1983, oh wait, let me go back to Peters. Peters in 1968, he, in Mexico, Peters documented the male of one of them in the nest with females of the other one. And so since he saw the male of one in the nest with females of the other type, he inferred that they're the same species. And, why would you have males and females of different species in the same nest if that weren't the case? But Thorpe came along in 1983 and said, no, I think they're different species. Williams in 2014 wrote a book called The Bumblebees of North America, an Identification Guide. And he really threw a wrench in the gears because he said they're the same species. So scientists bumblebee experts have been waffling back and forth all this time. Williams, in his identification guide, cited some preliminary unpublished genetic data and heard that there were intermediate forms found in Texas, morphological intermediates, and then some preliminary unpublished genetic data that suggested the same species. Bracero just very recently studied the chemistry of glandular secretions, secretions that are used to attract and recognize the opposite sex. And he found that the chemistry was different. Therefore, probably they're different species. So a few years ago, I got really involved in iNaturalist. And here on the right-hand side, you can see these are iNaturalist maps showing locations where people have observed the two species. So iNaturalist caused me to become interested in bumblebees. Before iNaturalist, a bumblebee was a black and yellow bee, and that's all I knew. Little did I know that there's two different kinds right underneath my nose. I hadn't even noticed. So iNaturalist brought this to my attention, and I got involved. Was fortunate enough to get involved with Jessica Beckham, who was a grad student at UNT at the time, and Jeff Johnson, who was her advisor. And I offered to sequence the mitochondrial DNA for them to add to their data set. And they're different genetically. So all of the ones that look like sonorous group together on one branch. And all of the ones that look like Pennsylvanicus, they group together on another branch. So they're monophyletic. They're easily recognizable genetically. This conflicts with Williams' preliminary unpublished data. So I think they made some mistakes in relying on unpublished preliminary data to draw taxonomic conclusions. But what I want to do right now is I want to back out just a second. I hope I'm doing okay on time. If I'm running out of time, somebody please tell me, but I'm almost done, like a couple of minutes here. I'm going to go out to iNaturalist. I hope everybody can see this okay. I'm on iNaturalist. Here's Georgetown. Somebody tell me if you can't see that. Here's Georgetown.
Yep, we can see it.
Okay, good. This is Bombus pennsylvanicus, the one that has the black between the wings and is the paler yellow. That's y'all's common bumblebee in that area there. And then if we look at the iNaturalist observations for Bombus sonorus, the one that's really bright, vivid, dark yellow with the yellow band between the wings, you can see nobody has yet documented them in was that Williams County, Georgetown area there, but just over there by Leander. So for those of y'all who are in the Georgetown area, they're knocking on your west door there, and I'd encourage y'all to be on the lookout for that darker yellow bumblebee with the yellow between the wings. They might occasionally wander out that far east. What I'm really interested in, and you can see if we go back and forth, there's places where they overlap. For example, there in the Leander area, west of Austin, the two species overlap. So what I'm interested in is, do they hybridize? Because some people have said they've seen intermediate forms indicating that they hybridize. And if they hybridize, we could question whether they're separate species or not. If they never hybridize, they are definitely different species, no questions beyond that. So what I'm doing, I'm collecting bumblebees where I'm collecting them from locations where you can find both species on the same bush. So they have the potential to hybridize if they want to. And then I'm going to look at their morphology. I'm going to look at their DNA, and I'm going to look for evidence of hybridization. So such evidence would be a bee that looks like Pennsylvanicus, but that falls out in the tree with sonorus. That would be evidence of hybridization. Okay, so you get your mitochondria from your mom. If your mom is one species and your dad is another species, then the DNA can get mixed up, and that'll show up genetically. So that's one of my current projects. I'm in the collection phase now, and I'm just now starting to get some DNA sequences, but I don't have any data to report from that yet. Okay, this is my last slide, so I'm going to answer my question: what is a species? Groups of organisms that have diverged sufficiently to be deemed worthy of recognition by biologists. Most scientists aren't going to like that definition. I like it because it reflects how scientists are actually doing things. Scientists may state that they're following a certain species concept, but there's other concepts that they could follow as well. How do they decide which concept to pick? How do you decide which criteria are most important? That's subjective. And all scientists need to realize that, right? So different people are going to disagree on what is worthy of recognition as a species. And that's kind of what makes science fun, I guess, is you can have an argument over a beer at a conference. Okay, it's always going to be subjective. Unless they never reproduce when they're together in their natural environment. That's really the only piece of evidence that is absolute. If groups of organisms are interacting in the same environment naturally, and they never reproduce with one another, no one is going to argue, no matter how many beers they've had, whether they're different species or not. But we don't always have that situation. So that's where the subjectivity comes in. Okay, that's it. I'm going to, any other questions? So does everybody see the questions, or do I need to read them?
You need to read them.
Okay. All right, so Jack posed a question here, a comment and then a question. So Williamson County appears to be a mixing zone for several different things, like dusky blue ground streaks, black crested and tufted titmice hybridized within that area also. So the question is, do any researchers do anything with these iNaturalist things that are only identified as general? In other words, you can't, you might not be able to recognize a organism as one species or the other because you're in a zone where they hybridize with one another. And how do scientists use the data that's that a lot of y'all are collecting out on iNaturalist? So iNaturalist is, as far as the scientific pursuit goes, we've been doing science since probably the 1600s or so, and iNaturalist has only been around a few years. So scientists, I think, are still learning how best to make use of citizen science data. For some organisms, it's very, very useful, particularly with these bumblebees. That's a great example, because as long as you get a decent picture, you can tell what species it is, basically. And so those maps I showed you, that's data that was totally unknown and unavailable to researchers just 3 years ago. 3 years ago, there weren't that many points on the map. It's been amazing just in the past 3 years to see the number of observations of those two bumblebee species accumulate and grow. And just probably within the past year, there's enough observations of that darker yellow bumblebee, Bombus sonorus, to be able to see that, gosh, it's really restricted by the Balcones escarpment, by the hill country. There's no observations at all once you move east off of the Balcones escarpment. And that's, I think, something scientists didn't know. And I don't know if any other bumblebee researchers are looking at that, but I'm definitely watching it. And I think here within probably a couple of years, somebody could definitely publish that pattern, that distributional pattern of bumblebees based on the iNaturalist data alone, because there weren't enough museum specimens to draw that firm conclusion that Bombus sonoris has a geographic distribution restricted by some kind of environmental change which maps perfectly to the Balcones escarpment. So that's just kind of one example of how researchers are starting to use the data. Okay, next question. Larry says, is a mule evidence that horse and donkey are the same or different species? All right, so horses and donkeys can reproduce. They can produce offspring that function very well. But most mules, the hybrid, cannot reproduce themselves. So there's a reproductive barrier, not in the first generation, but in the second generation. So since mules typically can't reproduce, we consider the horse and the donkey to be what some people call good species. They don't produce offspring that themselves can reproduce. Richard says, species are
all about the interpretation of a scientist.
All about the interpretation of the scientist. With one exception, if the species are living together and not reproducing, no one will argue there's no interpretation. They're definitely species. But if you can't determine if they're reproducing with one another or not, like the squirrels on the north rim of the Grand Canyon and the squirrels on the south rim of the Grand Canyon, there's no opportunity for them to reproduce. So we don't know if they could, if they had the opportunity. So we have to use indirect pieces of evidence to infer using our subjective criteria. So the answer to your question is usually species are all about the interpretation of the scientist. I would say yes. Some arguments are stronger than others. Charles and Judy, where do Denisovans fit into my discussion of humanoid species? All right, so Denisovans is this mysterious human, type of human that lived at the same time as the Neanderthals, but they occupied Asia, Eastern Asia. And Denisovans are probably more closely related to Neanderthals than either is to us. But we don't know that 100% for sure yet, I don't think. But the three of us, Denisovans, Neanderthals, and humans, the three of us are either members of the same species or different subspecies or varieties of humans. Again, it's subjective. It doesn't really even matter. They're slightly different. Who really cares if they're different species or not? The more interesting thing is that we've hybridized with all three. So people of Asian ancestry, especially Southeast Asians, have a lot of Denisovan DNA because they hybridized in the past in that area. Okay, so Karen asks, if a queen of one bee species mates with a male of the other species, won't the mitochondrial DNA just look like hers, not his? Yes. So how will mitochondrial DNA show hybridization since it's only coming from one parent? So my initial step is to compare the mitochondrial DNA with the morphology. So if the morphology is the pale yellow one, but the mitochondria is that of the dark yellow one, that would indicate hybridization. But I can also look at the nuclear DNA, because the nuclear DNA comes from both parents. And so if that bee is a hybrid, half of its nuclear DNA should be one species, half should be the other. And we can easily distinguish that with the genetic techniques. Marianne talks about ducks that hybridize. Ducks are crazy when it comes to species. Lots of different species of ducks can hybridize, and they do it a lot. And it drives the duck taxonomists insane. And that's just the reality. Some species don't care that they're mating with another species. Richard says, do you know about the 100th meridian moving to the east due to global heating? U.S. Department of Agriculture has this theory that the hill country will look like Laredo in 50 years. Have you heard this? It's a little bit outside my area of expertise, but if things are drying out and warming up, and they certainly are in my neck of the woods, then and we're seeing evidence with this in terms of the distribution of species. So this is how the two tie together. We are seeing some species now that didn't occur here previously. So as the environment changes, species that used to be unable to live in a certain area are now able to live in that area, and so their distribution changes. And so we're seeing a lot of insects that were never found north of Mexico that are now found up in San Antonio, for example. So we're definitely seeing the impact on climate change on species distributions. Have I seen, so Jack asks, have I seen the sturtlefish, the hybrid of sturgeon and paddlefish that scientists created? Not sure if it can reproduce, but what are the ethics of creating species or hybrids in the lab? Any rules for doing that type of work? Anytime you mess around with animals, especially their genetics, there's definitely some ethical questions involved. And researchers now have to jump through a lot of hoops to get approval to do their research. So we have committees that look at our research proposals and determine if that's something that the committee at least thinks should be done or not done. So it varies with the committee. There's informal rules and people whose job it is to think about these things. Okay, I got three more questions. Does the length of the line on the genetic tree mean anything? It depends on the tree. Sometimes the length of the branch on the tree tells you something, and sometimes it doesn't. On the trees that I showed, the length of the line was how different they are genetically. So the longer the line, the more mutations that particular organism has. So two species that are connected with one another by long branches, those two species would be very, very different genetically from one another. Whereas if they're connected by a short branch, they would be very similar genetically. All right, so Richard follows up with his question that he was thinking of species will have to adapt. So when the environment changes, in an organism, so all species have limits as to what type of environment they can live in. And they can't live in places outside of those limits. So when the environment changes, there's three things that might happen. If the species is mobile enough, to keep up with the changing environment, then that species, the individuals can move, right? They can move over many generations, but the range can shift from the north to the south or east to west. So the range or distribution can change as the environment changes. The second possibility is, and this happens when the organism cannot move or cannot move fast enough. It goes extinct. The third is adaptation. So adaptation requires genetic changes over many generations. And so if the environment changes rapidly, faster than what mutations can add new traits, then the species will go extinct. And so that's the problem, is the environment is currently changing faster than any other time in the history of life on the planet as far as we know. Scientists are able to track climatic changes indirectly with geology and chemistry and ice cores and things like that, tree rings, all kinds of indirect pieces of evidence of what the past environment was like. The environment's changing faster than it's ever happened before, as far as we know. So will organisms be able to adapt to that rapid change? Probably most species won't be able to. So we're seeing a lot of species go extinct now because of that. Extinction rate is higher than it's ever been documented as well, unfortunately. And Marianne follows up with the question, are the hybridized ducks able to reproduce? Yes. Maybe not every species, but like the mallard and the black duck, their offspring are fertile and can reproduce. They're not like mules that are sterile. But other species of ducks might hybridize and their offspring might not be able to reproduce because there's so many different species of ducks and I'm not familiar with all of them. Okay, I've run out of questions and I think I've run out of time.
Yes, you have. And thank you so much. We really appreciate you participating and taking your time to spend the evening with us. It's not an easy thing to do, especially over the computer. So thank you very much. And everybody have a good evening.